Imagine going back to a world where a simple paper cut could kill you, because that's really the frightening trajectory that we're currently on, where antibiotic resistance is set to surpass cancer deaths by the year 2050. So we can keep chasing our tails and making new antibiotics and then watching them, the pathogens gain resistance, or we can save some of these antibiotics and look to more innovative solutions. So most antibiotics are derived from microbes, and the microbiome approach allows us to treat the infection locally, thereby reducing side effects and toxicity. But the problem is that a lot of these therapeutic microbes can't survive inside the human body, or the ones that can't survive during illness or when the pathogen adapts. So SciBac's solution is to create better bugs as drugs using our DRIVE platform. DRIVE is a new non-GMO way to improve beneficial strains of microbes. It allows us to mate different species of bacteria or yeast to create hybrid strains. So much like you create a hybrid plant like a Meyer lemon, where you combine a mandarin orange and a traditional lemon to make a sweeter lemon. Or it's how you would make a mule, where you take the patience and the strength of a donkey and you combine it with the athleticism and the speed of a horse. So what SciBac uses DRIVE for is to move survivability traits and therapeutic traits from beneficial bacteria into safe probiotic strains. So like the mule, our hybrid strains have all the best traits from both parents. So in this example, we have a hybrid strain that can survive both a high fever and low protein conditions. So our first indication using DRIVE is to treat C. diff. And C. diff is a type of deadly diarrhea that is often contracted in hospitals due to spores. Spores are very difficult to kill, and they can withstand very harsh conditions and cause a recurrence of the disease. And in fact, 25% of these patients have a recurrence of the disease, and then those spores are easily spread in healthcare facilities. So SciBac's solution, what we have, our Difficil solution, is a 3 hybrid strain therapeutic that's taken orally, and we've designed it to meet all the problems that the disease causes. So we have 2 different lactobacillus hybrids that can kill C. diff directly, and they also prevent C. diff from binding to the colon. And then we have a 3rd hybrid. This one's a yeast, and it can neutralize the toxins that C. diff makes. And the whole formula prevents spore formation to end recurrence and stop the spread in hospitals. Our second indication is for chronic Pseudomonas infections in cystic fibrosis patients. By the time these patients are 18, 80% of them have a chronic Pseudomonas infection that hides in biofilm, and 90% of these patients die from this infection. So SciBac has created a single hybrid strain inhalable therapeutic that not only can eat biofilm and thin the mucus, but can directly kill the Pseudomonas where it's hiding. So SciBac's a different kind of microbiome company. Most companies, down there in the left-hand corner, they are banking on correlation being causation, and they're using wild-type or non-modified organisms as therapeutics. And then there are a handful of companies that are using traditional genetic engineering in order to overexpress a certain enzyme. But SciBac's the only company that can move multiple modes of action or mechanism into their therapeutics, into our microbes, and we can use any species that we want so that we can increase our efficacy and reduce the chance of resistance. So SciBac's founders are strain improvement and fermentation experts, and we are the inventors of our DRIVE technology, for which we have a wholly owned and issued US patent. We are using Todd Lorenz right now as a contractor for our clinical work. He brings 30 years of clinical experience, and we will be bringing him on as CMO at Series A. We have also hired regulatory and clinical manufacturing experts to ensure our success. Our scientific advisory board includes experts in the area of clinical C. diff, in gene transfer, antibiotic resistance, and in drug development. So I'm here today because we've got outside funding from Breakout Labs, and they are providing our matching funds for a $2.2 million CARB-X grant that's in its last stages of the application. And in addition to that, we have completed initial safety tests in mice in Stanford, and we have over $500,000 in revenue. This comes from pilot strain improvement projects. We have 2 of these going with large industrial companies, including Novozymes. So we are currently raising a $5 million Series A. This money will get us through an IND issuance as well as the completion of a phase 1 clinical trial. So please come visit myself and our CTO, Anthony, upstairs, because we would love to talk more science with you.